Hello, anatomy students. It is a rainy and dark Wednesday here in Somerset at about 3 p.m. I wanted to bring you a quick 20, maybe 25-minute lecture here that helps anybody who's missed class in the last 2 weeks get caught up. And then it's going to help everybody to kind of wrap up and summarize some of the more difficult concepts that we glossed over last week, specifically the action potential, things like wave summation and types of muscle movements like the twitch. I will cover those fast and slow twitch muscle fibers. Hey, how's it going? Here's my slide, and here's me on a slide. So I won't be covering, the skeletal muscle physiology, what's going on inside the sarcomere too much. And I won't be covering some of the basics like functions of the muscular system or types of muscle tissue. Those things I've offloaded to help you on McGraw-Hill with those virtual labs, those 2 virtual labs. What I am going to talk about are the action potential and how that signal gets from the neuron down to the neuromuscular junction. Some of the channels in the cell membrane that allow ions in and out. We'll talk about energy sources used in muscle fatigue. So there's this aerobic and anaerobic respiration, cell respiration that we talked about last Friday. I'm going to expand on that. And then I'll finally talk about types of muscle movements, fast twitch, slow twitch, and help us make sense of some of those graphs with voltage on them throughout this chapter. Okay, that's my intro. I'm going to pause here. Can you hear me now? Yeah, I can hear something now. All right. One small piece of anatomy I wanted to call out here was connective tissue. So I'll start at the bottom picture here. Let me get my laser pointer. These are individual muscle cells, also known as myofibers. Okay, those 2 terms, you can kind of exchange them without worrying about it. And what surrounds an individual muscle cell is the endomysium. That's the type of connective tissue that goes around just individual muscle cells. When you get a bunch of cells together in a little bundle, like they have in the second picture up here, you have a fascicle. And the fascicle is just a bundle of muscle cells, muscle fibers. You can see that connective tissue in between the perimysium. And then endomysium is the one that surrounds each individual muscle cell. Perimysium surrounds bundles of those cells, and then the epimysium covers the whole muscle. And you have your arteries and your nerves and your veins anchored within that connective tissue. You can see here. And then if we were to go lower, we would get back to those 2 myofilaments, actin and myosin. Myosin is the thick filament with the club head on it, and actin is the thin one. And this part right here. So this T-tubule, this green item here, helps conduct that electrical signal that releases ions. It's just infolding or an extension of the cell membrane here we call the sarcolemma. And then the sarcoplasmic reticulum, this webbed-looking thing here, that stores calcium. And that calcium, when it's stimulated, can be released all at once, causing this sarcomere to shorten and this sarcomere to shorten all at the same time and in unison. And here we have our little mitochondria. We'll talk about in a little bit the powerhouses of the cell. The neuromuscular junction, we've talked about what's involved here somewhat. This is the neurotransmitter acetylcholine. These are ligand-gated protein channels. And what I really want to focus on today is what's happening before you get down here to this section, the action potential. And that's where we're going to go from here. So action potential and muscle contraction. First off, we want to make sure that we know what a cell is doing, or what's the conditions for the cell and its environment when there's no movement, no message being sent down the axon to the muscle. Take note of these positive and negative signs here. The general environment outside of a cell, period, whether it's a neuron or another type of cell, is generally positively charged. Also, sodium, this Na, sodium is generally more abundant and more concentrated outside of cells in the extracellular space. If you cross your cell membrane and go down into the inside of the cell, the intercellular, this is where the cytosol is and the jelly of the cell, there's going to be an overall negative charge to this environment. And that's because there's all kinds of molecules. Cells are packed full of things like folded proteins, large protein molecules that are negatively charged. And then the other ion, potassium here, that is also most abundant or concentrated inside of the cell. Now there's still some sodium down in here, and there's still some potassium up in here in this environment, but it's not enough to make a difference. But what's established is a concentration gradient to where sodium is concentrated out here and it's very sparse in the cell. And if I want to pump sodium out, back up to those other sodium ions outside the cell, it's going to take energy. I'm going to use some of that ATP molecule energy to do that. And it goes the same for potassium being pumped into the cell. It takes energy to push things uphill. But if things are just at where they're supposed to be, you have a resting membrane potential, which is a certain amount of voltage or threshold that can easily be changed. And when it does, it propagates an action potential that travels down the axon to the muscle. Before we talk about how that happens, let's talk about how the membrane resting potential is just maintained. How do those concentration gradients stay the way they're supposed to are before we need them to flip-flop and make a change in the charge to propagate that? And so there's tiny little leak channels here. Some sodium can move through those passively. You don't have to have any energy to pump them through. And there's also some leak channels for potassium as well. And then there's this awesome membrane protein called the sodium potassium pump. It requires ATP to work, and it is pumping against the concentration gradients. And it so happens that it will not activate unless there's 3 sodium in there and 2 potassium in there. But once that happens, it will turn on and poof, pump them where they're supposed to go. And that's the way it goes every time. 3 sodium ions out of the cell, 2 potassium ions into the cell with the work of with the energy of ATP. And that's how it's going to, make sure that environment is maintained. And it's also how it's going to help reset too once the action potential has went through this area of the membrane. This just summarizes everything I've talked about so far. So as we talked about the resting membrane potential, what things are like before there's a signal sent down to the muscle cell. And when that happens, let me get to the next slide here. Okay, so I'm going to go back and forth between these two. Everybody's familiar with this graph so far if you've been in my class. This is the resting membrane potential, a certain amount of voltage for the cell membrane at rest. But if a signal is propagated up here, we can pretend this is a cell in our nose that is activated when a certain molecule hits it. We get a smelling sense. Once that happens, things will change inside the cell, and we'll send a electrical charge down here. If it's strong enough, it will meet this threshold to where this sodium gated voltage gated channel will spring open and allow sodium ions to rush in. If you remember, it was positive on the outside, negative on the inside. But with that big rush of sodium ions going in there, it's going to become more negative on the outside. And then as that voltage has increased enough to meet another marker, the potassium voltage-gated ion channels can open and allow potassium to rush out of the cell. And so we get this swift exchange of ions and a swift exchange of charges on each side of the membrane, and that is called the action potential. So your job is to know where sodium and potassium lives normally, which way they're going during the event of one of these voltage-gated channels opening. And you should also know that these words depolarizing and repolarizing, what they mean and what's happening during that time. So as this cell is depolarizing, that charge is leaving or propagating out. That's the sodium channels opening as first. And then once it hits a certain voltage up here, potassium ions will open after that. They happen second. And then you get the repolarization phase where things are recharging or resetting for the next change. I think that pretty much covers it. Let's go to the next section here. All right, types of muscle contractions. These are two terms I went over quickly in class last week, but just to help anybody who's not familiar with them or help somebody recall them, make sure that you understand metric is like a meter. You're measuring something, and when it's ISO, it's not changing. It's isolated. So you can have a muscle do work. I can press on the wall and do an isotonic or isometric contraction, and it's like a newer way to work out. There's like some people that say, oh, that's better than lifting weights or doing concentric muscle contractions or eccentric. Anyways, isotonic means that there's no change. It's isolated, the tonicity of the muscle. And so a muscle can change length. You know, my bicep got longer, tricep got shorter here. But the tension in the muscle did not move. I'm not flexing any harder than I was a minute ago, but there's those two things changing there. Just be familiar with those terms and little tricks that help. I just gave you to remember them. Signal frequency and intensity in muscle contraction. So this is interesting. How do we know that when I go to pick up this light book, it's nothing I could sit here all day like this. I'm not having a real issue with any arm fatigue. But if I want to get this one and hold it up, it's a lot more work. And if I sit here and continue to maintain this posture, you know, my muscle's going to start to fatigue. I'm going to start getting tired. If you were to add another book on top of it, I would feel it. Where does it come from that I know to just lift a little bit on that one, but more on this one, and to maintain it? That's pretty neat to me. I'm like, I'll never be able to figure that out. But it is it is discoverable. We can learn it. So tension of the muscle and time here. Let's look at a couple slides here. Make sure you understand what frequency and amplitude are. These are like physics terms. Frequency refers to the intensity of the signal, or how many times the wave is coming in a period. So faster wave lengths or faster frequencies mean a lot more points here. You can also talk about a wave in terms of amplitude. How high is the wave? This is what we would call signal intensity or signal strength in terms of a muscle contracting. So these are wider apart. They're at lower frequency. They're both at same amplitude. So this one's coming faster. This one's coming slower, and that's all. But if this was a higher wave, we would say it's a stronger signal. So frequency, you know, how many times is it happening? Is it happening fast or slow? That affects how our force is generated for our muscles to pick things up. In higher frequency, if the signal is coming faster, the action potential, then you get a stronger contraction. And if it's coming fast enough, that muscle fiber that's being contracted can get contracted to the point where it has no chance to reset, to relax, and that's known as summation for that muscle fiber cell. Okay, so increased frequency doesn't give the muscle cell or the sarcomere a chance to relax, and so it stays contracted. And the faster that happens, the more those tiny parts, the myosin, the actin, that's the less time they have to relax and reset, or for ions to be pumped in and out. Summation. The higher the frequency, the stronger and more sustained the contraction. So a rapid series of signals can lead to tetany. Hopefully we have a little bit of understanding what that word is. Continuous maximal contraction, and thus maximum force in that particular muscle fiber. Intensity, or height of the wave, amplitude, if it's more intense, you know, you can't really do anything else with those muscle fibers that are maxed out. But what you can do is increase the sound, increase the call for help, and recruit more motor units together to help lift this thing. And so once you get more motor units moving, you get more muscle force contraction out of a greater signal intensity. Just to make sure we know what a motor unit is, hopefully I have some good depictions of them here. These are muscle fibers, and we'll say this one axon, this one neuron here is connected to this unit, and that's all it will recruit. when it's called on by my brain. And to lift this real quick, I only need a few of those. Okay, the same ones might be involved whenever I'm writing in my book. If I try to do that with something more heavier, or if I'm doing it for long periods of time, I'm going to need more help, and these guys that were working initially aren't going to cut it. They were activated during the twitch movement. Their threshold was met. It's contracting. But if I sustain that force and my signal gets stronger, see my amplitude getting taller here? More motor units are going to be recruited, and I'm going to get more lifting power. What's key is that these are activated at a certain voltage threshold, and these won't be activated until more voltage is met or the signal got more strong and they were able to meet their thresholds and jump in there and help. Okay. So twitch, this is the initial quick movements. You might be able to do them for a long period of time, but if you're going to lift anything heavier, the more weight you add, the more motor units that are recruited, and you get something called wave summation. So we talked about summation of a fiber, muscle fiber. Now we're talking about wave summation and multiple motor units being recruited together. If I were to stand here and hold that out, I think it would be considered incomplete tetanus because my muscle is contracted, but it's not as contracted as it could get. So it's incomplete tetanus. Think about the muscles that allow us to sit up and hold our head up without thinking about it. They are incomplete tetanus. And the complete tetanus is everything is just at max, going all out. You can see that here on this slide. And so you can use all these sometimes. You can use one or a few of them at certain times, depends on what's going on. These next 4 slides hopefully will illustrate some practical applications of this. A light object, initial force by your muscle is strong enough for that little contraction to pick up the pen or the book or to write. And then if you need more force, you need more lifting power, you start to recruit more motor units around those initial twitch motor units. And then if you keep going, you get tetanus. Okay, let's pause there for the next section. Okay, let's talk about energy sources, muscle fatigue, and things like that for just a minute. Hopefully everybody knows about ATP by now. It's if glucose is the molecule that we need to survive, primary sugar in most of our food, and a cell needs it to do work, they have to break that glucose molecule down into something that can be put into their machine. It's like a $20 bill. You can't put that in a vending machine. You have to break it into quarters. ATP is the quarter here. Another molecule that can help generate ATP that's not glucose is creatine phosphate. It's particularly cool in the muscles. Creatine kinase is an enzyme that breaks that down, and we can test for muscle damage with that enzyme. You can get an idea how your kidneys are working and things like that. And these energy sources can be used to make ATP in 2 different types of environments: a rich oxygen environment or a low oxygen environment. So anaerobic. Oh yeah, this is important too, this little organelle, the mitochondria, the powerhouse of the cell. These processes will be happening inside of the cell or outside the cell, depending on the situation. We'll talk about that in a second. Okay, immediate energy. Your muscles are full of creatine phosphate, and that holds a lot of energy. Creatine phosphate will put a third phosphate to ADP, adenosine diphosphate, making it adenosine triphosphate. Where's my phone ringing? So this is for a short burst of energy. You only have a little bit of it. It didn't last that long. If you were to get up and run out of here, if I were to get up and run out of my office, I would be able to run pretty good for about 15 seconds to 2 minutes. But soon the oxygen that was just hanging out in my cells in these areas that we're talking about for these chemical reactions, that would be used up really quick. And then I would need to replace that with my blood flow. Once it runs out, once that O2 is used up, we're now in an anaerobic condition or anaerobic respiration where the cell will be making energy via ATP, making ATP in an environment where there's no oxygen or very little. One product of this anaerobic respiration is lactic acid. And normally lactic acid would be changed back to lactate in the blink of an eye or vice versa. But when it's in the no O2 or anaerobic environment, it's going to stay and hang out and it's going to build up. And when that happens for too long, if I were to hold this book up forever, my muscle would start to burn. And that burning is directly caused by the lactic acid buildup. And so too much lactic acid, muscle quits working. That's one thing that leads to fatigue, other than a few other things we'll mention here in a second. Okay, once I ran out of here and was running for about 2 minutes, I would then switch over to aerobic respiration or oxygen-rich environment cellular respiration. You can equate aerobic with long-term endurance type stuff. Oxygen is required in this way to produce ATP. And it's the one that's good for several minutes to hours. So up here we have our glucose molecule. Let's just say it has been taken into the cell. And we need, there's enzymes that's going to start working on it to produce 2 ATP. So you get 2 out of that for whatever work you need to do initially out here in the cytosol. You also have to put 2 in too. So we have to make sure we're not spending more than we make, right? Glucose is broken down and changed into this these 2 3-carbon molecules here. And then if it's an aerobic environment, if I've been doing that for a while, that pyruvate can move into the mitochondria where a bunch more chemical reactions happen. And I will get some ATP out of it again. But it's just not as efficient as if there is oxygen present. So if glucose is happening, it's aerobic conditions, you can just move right over here. And so the products of this first reaction, these electron acceptors here, or electron or proton donors, can go in here to the electron transport chain and make ATP way more efficiently. Look at this payout down here, 28 versus 2. 28 versus 2. And this is what's going on, between aerobic and anaerobic, and why you have to switch over. To make ATP down here in the mitochondria, here's that ATP synthase I highlighted for you all. It's one of my favorite proteins. You have to take these electrons that are doing work here and put them somewhere. And O2 is the best electron acceptor we got. That's why we require oxygen. That's why these are being made outside and sent in here. We're making electron acceptors out here. So, anaerobic, quick, there for us, not efficient in making ATP, doesn't last long, but aerobic conditions, the mitochondria can shine, the powerhouse of the cell, it can do great work and make efficient ATP for us and get a big payout from the glucose that we have to pay in. Yeah, that says it. So, along with lactic acid accumulation, what causes muscle fatigue? Depletion of ATP and the ion imbalance, the calcium, the sodium, the potassiums, those things have to get put back in place. And when there's too much work going on, they don't have time to do that. There's also this thing that is called oxygen deficit. So runners or some people who are working out will call this the afterburn effect. And I've really never experienced it. I used to be an avid runner back in the day. But the oxygen does have to be repaid. It just have to be repaid. That debt does have to be repaid before you can get back to normal metabolism and things like that. Let's see here. I think I got a marathon runner. Yeah, so the sprinter, he's anaerobic conditions, but he didn't need much, right? He's only going to be running for just a short period of time. The marathon runner, though, he's in it for the long game. He's going to be mostly doing aerobic respiration, O2 rich environment, efficient ATP production. Mitochondria are getting to work with your cells. Okay, hopefully that helps clear those two things up. So, between humans, you can't really differentiate between slow and twitch, fast twitch and slow twitch. But an athlete who has trained for many years and people have said, oh, he's good at sprinting. He might have more slow twitch fibers built into his DNA and to his body than other people might. And he's been training and really intensely focusing on recruiting those motor units of those fast slow twitch endurance things. Might have got that just flip-flopped. So we all have an uncertain amount balance between it. Nobody is really born with more of one type than the other, but some people are genetically predisposed to be a little bit better at one of the two, and if they train those and develop those parts of their body, then they can call on them a lot better and be great sprinters or great marathon runners. Slow twitch muscle fibers. Remember we talked about switch earlier, or twitch with the book. Okay, slow twitch and fast twitch. Slow twitch are adapted for endurance. Okay. This protein called myoglobin, which is just sort of like hemoglobin's cousin in terms of storing oxygen, that is going to be really abundant in slow twitch muscle fibers. And that makes sense. Aerobic conditions requires oxygen. We need stores of oxygen everywhere. So myoglobin serves that purpose. Fast twitch muscle fibers are adapted for quick, powerful movements. They typically are in anaerobic conditions, so you're not going to see a whole lot of myoglobin in there. Still be some. Here's some practical examples again to hopefully help illustrate these concepts. Imagine you're going to pick up your pen and write an assignment out. So that would be slow twitch. Okay. Endurance. Your hand might get tired after a few hours of writing, but it can hang in there. Myoglobin's role, I've already kind of stated that. If you're going down the stairs, for example, just imagine you're trying to get down quiet or something, and you're controlling your large, massive thigh muscles and your trunk and everything. And they're partially contracted, partial tetany, but you slip and you're like, oh. Then your fast twitch are going to be engaged as well. They're going to be using what we stated is just pretty much normal conditions for muscle cells and what they have stored and what they use up, what they do after those things are gone. Finally, this is why we have dark meat, light meat, and birds. Think about a chicken, which is all, usually light meat, white meat. Chickens can't fly that far. They can only flap and then they're done. But ducks, maybe even turkeys, turkeys don't really fly that far. Ducks do, and they have a lot of dark meat, especially in the breast area, like chickens, that we tend to enjoy more. At least I do. So if it's slow twitch, high endurance, it's going to have that myoglobin molecule in there, and it's going to make it darker in color. You can't really differentiate between fast and slow twitch fibers in human beings. All of ours just looks the same. All right, that's the end of it. I hope I haven't rambled on too far. Hopefully I've helped you to understand a few concepts here and help you to get a little more confidence in what you're studying. I look forward to seeing you all on this Friday. Have a great afternoon.